Hey everyone, it's Sheardeeh and this is Biology 101. In today's vlog, we're going to be talking about osmosis. Now, what is osmosis? Well, osmosis can be defined as a process in which molecules of a solvent pass through a semi-permeable membrane from a lower concentration to a more concentrated one, thus reaching equilibrium between each side of the membrane. Now, how do we achieve this? Well, we can do a simple experiment to understand how osmosis works.
To do this, we will need salt, a graduated cylinder, some water, 2 glasses, a cutting board, a spoon, a carrot, a steering rod, and a knife. First, we will cut the carrot into 2 pieces on the cutting board. Reminder to be cautious in handling a knife. If you are incapable of using one, you may ask someone else to assist you. Then, prepare 2 glasses and fill them with 150 milliliters of water using the graduated cylinder. Take 3 tablespoons of salt and pour it on one of the glasses. Then, use the steering rod to efficiently dissolve the salt in the water. Finally, place the cut-up carrots in the glasses. As osmosis occurs, let me tell you what's happening in the solutions. Carrots are made of cells, and their cell walls act as semi-permeable membranes. In the cell, water is less while the solution outside has more water. In order to balance these concentrations, the water from the glass moves into the carrot cells, pushing the cell walls and making it bigger and more rigid. Meanwhile, in the salt solution, it is the opposite. The salt concentration in the glass is higher than that of the carrot cells. This is why the carrot appears smaller and becomes less rigid. The solute concentration is greater outside the cell than inside of it. Now, we will take out the carrots. Placing them on a tissue paper to dry, we will now examine the difference between them. Taking the carrot from the salt solution, we can see that it is easily bent. As we take the carrot from the one submerged in water, we can observe that it is very rigid and hard to the touch. In the side-by-side comparison, we can see that indeed the carrots submerged in the salt solution appear smaller and less rigid than the ones submerged in water. We can conclude that the carrot submerged in the salt solution has the tonicity of a hypertonic solution. Since the solute cannot pass the cell membrane, water can. Hence, it will flow outside of the cell in an attempt to reach concentration, equilibrium, or homeostasis between inside and outside of the cell. Its net water movement is out of the cell, and the end result is crenation or shrinkage of the cell, making it less rigid. On the other hand, the carrot submerged in water has the tonicity of a hypotonic solution. The concentration of solute is greater inside the cell than outside of it. Water will flow inside the cell in order to reach homeostasis. Its net water movement is inside the cell, and the end result is the swelling of the cell or hemolysis.
How did you find the experiment? Was it too hard or was it too simple? Well, the important thing is you understood how osmosis works. To summarize, in osmosis, water moves from areas of low salt concentration to areas of high salt concentrations. Once we added salt to the water, we created a higher salt concentration, which made the saltwater carrot softer than the regular one. Did you find that interesting? Because I certainly did. Well, thanks for watching, and I'll see you in the next one.